Hi, welcome to my poster titled Cortical Atrophy Signatures and Machine Learning MR-Based Classification of Primary Progressive Aphasia. PPA is a rare neurodegenerative condition characterized by gradual impairment in language processing. There are three main clinical subtypes of PPA: the semantic, logopenic, and non-fluent variants. These three variants have relatively distinct language deficits, particularly at early stages of the syndrome. Importantly, individuals from each of these subtypes often present with cortical atrophy in areas associated with the specific language domain that is compromised. For a brief summary of these PPA variant characteristics, please refer to the table in this background section. Broadly, the goals of this study are to outline the signatures of each of the three PPA subgroups. To do this, we employ two methods. We'll first use univariate methods to validate and further expand upon atrophy characteristics within and across PPA variants. And then secondly, we'll focus on classification, which we perform on data that is reduced via principal component analysis to identify differential regions across the PPA sample. We obtained a balanced PPA sample of 20 individuals per variant, and for more information about the sample demographics and exclusion criteria, please review this sample section. Shown in the language data section are the scores obtained using the Progressive Aphasia Severity Scale. You can see here that our patient sample consists of patients that are in the early stages of the syndrome, and that the three variants have relatively disparate language deficits. To identify group atrophy signatures, we computed W-maps to control for age and sex for each individual in the PPA cohort. Please refer to the formula provided to get a sense of how these values were computed on the surface maps. Represented below are the mean W-maps per PPA group. As you can see, the semantic group shows the highest magnitude, suggesting either more atrophy across the semantic patients or more group homogeneity in terms of atrophy location. Next, we look at the regional overlap of the top 10% of individual W-maps per cohort. This method equally weighs each individual W-map, controlling for different W-score ranges across subjects. Here you can see the most common location of peak atrophy across patients for a given PPA group. We can see that the semantic PPA group, again, shows the highest degree of overlap, suggesting this group is more homogeneous in its atrophy presentation compared to the other two variants. Here we shift our focus to classification by first identifying regions that best differentiate the three cohorts. Using principal component analysis, we derive the following eigenbranes from the left hemisphere W-maps. These three eigenbranes capture approximately 50% of the variance in the entire PPA sample, and you can see that the positive values on these maps isolate regions that correspond to a unique PPA cohort, demonstrated by the negative PC weights shown in their adjacent histograms. We then use the weight of each subject for the first three eigenbranes to then train a support vector machine classifier using the k-fold cross-validation method. We found that classification performed with relatively high accuracy, ranked by performance in this table. The plot to the right is the 3D representation of the entire set of the first three PC weights. And to conclude, we have shown that each PPA subtype has distinct cortical atrophy characteristics, and that using multivariate methods, we demonstrate cortical areas important for differentiating the three subgroups. And we find that the three largest sources of variance correspond to the three subgroups in the sample. Lastly, using the principal component weights, an SVM classifier can be trained to predict PPA subgroup. Thank you for coming to my poster presentation. I also want to thank my colleagues and co-authors on this poster for their contributions to this work.